Chapter 12 Mineral Nutrition. The basic needs of living organisms are essentially the same. They require macromolecules such as carbohydrates, protein, and fat, and water and minerals for their growth and development. This chapter focuses mainly on inorganic plant nutrition, wherein you will study the method to identify elements essential to growth and development of plant and the criteria for stabilizing the essentiality. You will also study the role of the essential elements, their major deficiency symptoms, and the mechanism of absorption of these essential elements. The chapter also introduces you briefly to the significance and the mechanism of biological nitrogen fixation. 12.1 Method to study the minerals requirements of plants. In 1860, Julius von Sack, a prominent German botanist, demonstrated for the first time that plant could be grown to maturity in a defined nutrient solution in complete absence of soil. This technique of growing plant in a nutrient solution is known as hydroponics. Since then, a number of improvised methods have been employed to try and determine the mineral nutrition essential for plants. The essence of all these methods involves the culture of plant in a soil-free, defined mineral solution. These methods require purified water and minerals nutrition salt. Can you explain why this is so essential? After a series of experiments in which the root of the plant were immersed in nutrition solution and wherein an element was added, removed, or given in varied concentration, a mineral solution suitable for the plant growth was obtained by the method. Essential elements were identified and their deficiency symptom discovered. Hydroponics has been successfully employed as a technique for the commercial production of vegetables such as tomato, seedless, cucumber, and lettuce. It must be emphasized that the nutrition solution must be adequately aerated to obtain the optimum growth. What would happen if solution were poorly aerated? Diagrammatic view of the hydroponic technique is given in figure 12.1 and 12.2. 12.2 Essential mineral elements. Most of the minerals present in soil can enter plants through roots. In fact, more than 60 elements of the 105 discovered so far are found in different plants. Some plant species accumulate selenium, some other gold, while some plant growing near nuclear test site take up radioactive strontium. There are techniques that are able to detect the mineral even at a very low concentration, 10 to the power minus 8 g per ml. The question is whether all the diverse mineral elements present in a plant, for example gold and selenium as mentioned above, are really necessary for plants. How do we decide what is essential for plants and what is not? 12.2.1 Criteria for essentiality. The criteria for essentiality of an element are given below. A. The element must be absolutely necessary from supporting normal growth and reproduction. In the absence of the element, the plant do not complete their life cycle or set off the seed. B. The requirement of the element must be specific and not replicable by another element. In other words, deficiency of any one element cannot be met by supplying some other elements. The element must be directly involved in the metabolism of the plant. Based upon the above criteria, only a few elements have been found to be absolutely essential for plant growth and metabolism. These elements are further divided into two broad categories based on their quantitative requirements. 1. Macronutrients and 2. Micronutrients. Macronutrients are generally present in plant tissue in large amounts in excess of 10 mmol kg of dry matter. The macronutrients include carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, potassium, calcium, and magnesium. Of these, carbon, hydrogen, and oxygen are mainly obtained from carbon dioxide and water, while the other are absorbed from the soil as mineral nutrition. Micronutrients are trace elements are needed in very small amount, less than 10 mol kg of dry matter. These include iron, manganese, copper, molybdenum, zinc, boron, chlorine, and nickel. In addition to the 17 essential elements named above, there are some beneficial elements such as sodium, silicon, cobalt, and selenium. They are required by higher plants. Essential elements can also be grouped into four broad categories on the basis of the diverse function. These categories are 1. Essential elements as component of biomolecules and hence structural element of cell. Example carbon, hydrogen, oxygen and nitrogen. 2. Essential elements that are components of energy related chemical compounds in plants. Example magnesium in chlorophyll and phosphorus in ATP. 3. Essential elements that active or inhibit enzyme. For example, magnesium 2 plus is an activator for both tribulose bisphosphate, carboxylase, oxynase, and phosphophenol pyruvate carboxylase, both of which are critical enzymes in photosynthetic carbon fixation. Zinc 2 plus is an activator of alcohol dehydrogenase and molybdenum of nitrogenase during nitrogen metabolism. Can you name a few more elements that fall in this category? For this, you will need to recollect some of the biochemical pathway you have studied earlier. 4. Some essential elements can alter the osmotic potential of a cell. Potassium plays an important role in the opening and closing of stomata. You may recall the role of minerals as solutes in determining the water potential of a cell. 12.2.2 Role of macro and micronutrients. Essential elements perform several functions. They participate in various metabolic processes in the plant cell, such as permeability of cell membrane, maintenance of osmotic concentration of cell air, electron transport system, buffering action, enzymatic activity, and act as major constituent macromolecules and coenzymes. Various forms of and function of essential nutrient elements are given below. Nitrogen. This is the essential nutrient element required by plant in the greatest amount. It is absorbed by an NO3 minus, though some are also taken up as NO2 minus or NH4 plus. Nitrogen is required by all parts of a plant, particularly the meristematic tissue and the metabolically active cell. Nitrogen is one of the major constituents of protein, nucleic acid, vitamin and hormone. Phosphorus. Phosphorus is absorbed by the plant from soil in the form of phosphate ion, either as H2PO4 minus HPO4 2 minus. Phosphorus is a constituent of cell membrane, certain protein, all nucleic acid and nucleotide, and is required for all phosphorylation. reaction. Potassium. It is absorbed as potassium ion K plus in plant. This is required is more abundant quantities in the meristematic tissue. Buds, leaf and root tips. Potassium help to maintain an anion-cation balance. Cell N is involved in protein synthesis, opening and closing of stomata, activation of enzymes, and in the maintenance of the turgidity of cells.